So, hello everyone. We are here once more in our section of what we are reading of the South American Mycorrhizal Research Network YouTube channel. And today we are lucky enough to have Johanna Wong from the Elizabeth MacArthur Agricultural Institute in Australia. This is a government institution for agricultural research. In addition, Johanna was recently awarded in the ICOM11 meeting, an international conference of mycorrhiza meeting, as a student award in excellent mycorrhiza research. So I will show you to all of you the paper that had this award. So her paper was called the ectomycorrhizal fungus Pisolithus microcarpus encodes a microRNA involved in cross-kingdom gene silencing during symbiosis. So this was published this year in PNAS. Her Ph.D. advisor was Jonathan Plett. So hello, Johanna, how are you?
I'm good. Hi. Thank you very much for inviting me here.
Great. So tell me a little bit, like in general, how this research came about, and what was the main question behind this paper?
Sure. Actually, this research came about, the project itself, you might have to ask Jonathan himself because all I did was that during the time I saw that there is a PhD position opening in Jonathan's lab in Western Sydney University, and he's looking for a student to look at protein effectors and other molecular signals in ectomycorrhizal fungal interaction with eucalypts at the time. And at the time, I was a master's student that studied in Hong Kong. And I in my master's thesis, I was looking at the role of small RNA in soybean during the symbiosis of rhizobia. So I thought that, it would be very interesting if we actually look at small RNA in ectomycorrhizal fungi as well. And I have checked and then look at the internet to find if there are any publications in this area. And I found that during the time, small RNA is a very new area in ectomycorrhizal fungi, at least. There isn't any publication on it. So I thought I would ask Jonathan, hey, how about can we actually try with this idea, with this PhD project? That would be great because I have some experience in that. And he taken it on board. He's very open to this idea. So that's why we have started this project. And then we try to look at mainly 3 different questions, whether small RNA in fungi would play any role in the ectomycorrhizal fungi in eucalypts interactions, and whether there are cross-kingdom small RNA transfer between the 2 interacting parties, and what kind of role these small RNAs might be playing during their interaction. So that was what we do.
So you mentioned that the main topic was small RNA, sorry. So me not being like a mycorrhizal molecular mycorrhizologist, explain us a little bit more what exactly are small RNAs and how usually they work and why they have not been so much studied in ectomycorrhizal.
Sure. Small RNA in essence is actually just small RNA, as in RNA molecules that are small, because they are very short in length. They are usually around maybe 18 to 30 nucleotides long, around that. And they are not protein coding, so they won't be translated into protein. And what it does usually in an organism is that they can regulate gene expressions, and it will target the messenger RNA and inactivate their target's expression. So by doing that, then by binding to their target, then the target gene expression will be reduced. So this is a way that, in animal and plants are very well known during the time. And I think in fungi, relatively there are less study on it, but we are catching up on that too. So small RNA is an umbrella term that describing all different sort of small RNAs. It's a classification of that. And microRNA is a class of small RNA. There is also other classes of small RNA, for example, small interfering RNA, etc. So what microRNA is particularly interesting is that it is most well known of its hairpin structure of the precursor. So the precursor of a microRNA will be binding on both ends. So it's forming like a structure that looks like a hairpin. And that's how we can identify it or predict it using small RNA sequencing data. So it's more well known that microRNA is synthesized in animal and plants, but in fungi probably is a relatively less studied area.
Thanks. So tell us a little bit about, and I will share again the screen, a little bit more about the methods that you use. You had this really nice first figure, right? So
yeah.
Can you tell us what did here and how long did it take you to do the experiment itself?
So what we did mainly is looking at doing the small RNA sequencing at different time points during the ectomycorrhizal fungi symbiosis formation. And we mainly doing it in agar plates. So it's a very controlled environment rather than in the environment, I mean, in the natural environment, because it's easier to control what's happening, what kind of interactions is happening, and what time, I mean, what is the duration of the interaction. So that's why we do it in a Petri dish. And so that's one thing that particularly key to our discovery is a pre-symbiosis setup that is shown on the figure C over there. So what we did is we put a cellophane membrane in between the fungi and the plants so that they are physically separate from each other, but then the membrane is still have pores that are big enough for chemicals or molecules to be exchanged between the two parties.
Wow.
So after a week of this sort of interaction, we have then taken the roots of these plants and then do small RNA sequencing on it. And based on our analysis, we did identify these microRNA that are supposed to be synthesized by the fungi rather than the plant in these roots. And so as an additional confirmation, we have also done some confocal microscopy that are in the figure F2O over there. So what we have shown is that when we're using a probes that are binding to these microRNA, the probes could be fluorescence, right? So in these figures will be in the blue color. So when we look at those roots that are interacting with the ectomycorrhizal fungi, we can actually see these blue signals in the roots, which means that to us that these fungal microRNA are localized in the plant root during the interaction. So that actually gives us another layer of confirmation.
Of evidence, yeah. One quick question. You mentioned that you put this membrane. So what is the diameter of each? Like, how thin is this membrane?
How thin? I actually didn't really look at how thin they are, but as this figure D show over there is the EM microscopy picture. So we can actually see that these fungi are like physically separated from the below of the agar. So we know that the fungi are not because so cellophane is made of cellulose and ectomycorrhizal fungi cannot digest cellulose. So we know that they can grow and penetrate that.
Oh, great. And finally, here in figure A and B, when it says pre-symbiosis, this means 1 hour before or 2 hours before? What exactly that means?
So pre-symbiosis is in these two figures is actually meaning it's the same setup as the figure C, but we have only have it for 24 hours at that time point.
Okay, I see.
So what we were trying to simulate is that we want to simulate a time point that before the fungi go into physical contact with the root, and then in the 24 hours is the actual interaction where they are physically touching each other.
I think that you already mentioned like the main finding, but what do you think will be the take-home message of this? What is the main thing that you discovered here, and what is the implication of this discovery to future research?
I think it's a very exciting finding that we have, that we found that actually fungal microRNA could be transported from the fungi. So sorry, taking it back a little bit more actually. First of all, we find that ectomycorrhizal fungi can synthesize microRNA. That's new. And then second of all, these microRNA are transported from the fungi to the plant interacting partner. And furthermore, we find that these microRNA, if you read the paper, because besides we find where they are, we actually look at what the functions it is as well. So we do find that these microRNA could stabilize the symbiosis. So when we try to inhibit this microRNA's action, it actually can lead to abnormal mycorrhizal formation, and it can reduce the hartig net depth as well. So that's how we know that it actually play a role. Mm-hmm. So I found that this is very exciting because, I mean, ectomycorrhizal fungi interactions or mycorrhizal fungi interaction in general is very common in all sort of plants, maybe except brassicas and the very small amount of plants that we can't really see it. But that means that this sort of cross-kingdom small RNA transfer could possibly happen in a lot of these interactions. So yeah, it's very exciting.
Do you think that this is also common across two final questions, sorry. Do you think that this is common across other mycorrhizal types like arbuscular or ericoids or orchids? And second, because the experiment was controlled, because of course it needs to be controlled, but what will happen in nature, like under natural conditions with this process of microRNA transfer?
I think it's quite possible that it can happen in other kind of fungi, but I think I probably won't be the best person to answer it. I think I need someone that know more about evolution, why it's like how different all these different sort of mycorrhizal interactions could be, to know that whether it could be happening in other kind of interactions. But I myself would think that it is very positive that this could happen in other mycorrhizal types. And in a natural environment, that's given that I think it could happen when they are actually in physical contact, but before they come into physical contact, I mean, before the fungi come into physical contact from the root, I would think I would be quite, I don't know, hesitant to say that that could happen if, you know, just by diffusion to go from the fungi to the root through the soil pores. I think that would be quite hard to know, but just by thinking about it, there's so many different possibilities that could happen. And I think that we can possibly, for example, do some meta-transcriptomics to try to see what kind of small RNA there are in the soil, or just to look at how diverse that could happen, right? So I think that's very exciting.
Yeah, that's absolutely exciting. And also because I guess that many, of course, in nature, in the soil, many mycelium from many different species can be interacting, and maybe there is like some, I don't know, some phylogenetic differences among different ectomycorrhizal lineages. So this
Yeah.
It's super exciting. So
Yeah.
to finish, tell us what are you working right now? Which is your current position? What are you looking for in the future?
Oh, my current position actually diverted a little bit away from mycorrhizal fungi. So
Mm.
instead of looking on symbiosis, I'm actually looking at pathogens. So my current role is in the team of plant biosecurity, and I'm looking at developing molecular diagnostic tests for detecting plant pathogens and mainly bacteria. But
Mm.
I mean, looking into the future, I feel that besides we wanted to detect something from, like, for example, plant pathogen, because I feel like detection is actually like a reaction, right? But I also wanted my research to be more proactive. So looking at, I mean, mycorrhizal fungi is a very interesting area, and I want to continue on that too. So it would be great if I can look at, for example, how mycorrhizal fungi could help in defending like pathogens. So
Yeah.
that would be something to look at. And I guess as a molecular biologist, I would want to know, for example, if this small RNA could be transferred from a fungi to the pathogenic bacteria, like all
Oh,
sort of interactions that could happen, right?
super
Um,
interesting. Yeah.
yeah. So that's what I wanted to do.
Okay, so thank you very much for being here, and again congratulations on your award, very well deserved.
Thank you.
And thank you all, and see you next time.
Thank you. See ya.